There's a whole other story there that I don't have time to cover now, but I do just want to make a few comments on measuring of soil carbon and how we've gone about doing that and where we've ended up with this. There's a whole load of standard methods that people use. The ecologist's favourite, loss on ignition. Many of us would have spent lots of hours in the lab doing Walkley Black years ago. Now typically we're using dry combustion chromatography or LICOs or elementars. We can measure some sort of labile carbon using permanganate, oxidizable carbon. There's particulate organic carbon methods. I'll say something about that later. Various molecular analysis, I'll come back to that. And carbon isotopes, and I will show you some isotope data. And these are all increasingly, this increasing diversity is increasingly used, which often offers us some useful tools to look into carbon in a little bit more detail. There's also mid-infrared. I won't comment on that. The molecular level molecular level organic analysis is also worth saying a little bit about. There are some GCMS techniques where we can use biomarkers, particularly PLFA, which are used in microbial community ecology, but also we can try and understand a little bit more about the quality of organic carbon, and I'll come back to you on that. NMR, which is used commonly now, FTIR, LCMS, and compound-specific isotope analysis. Now, some of these are highly illuminating with specific projects, with specific questions or hypotheses to test within context. But because they're there doesn't mean we have to use them. We need to use the right tool for the right task. So make sure we try and keep the methods appropriate for what we need to do. So a relatively simple one that any lab can develop is a fractionation process where we fractionate soil according to its apparent lability or recalcitrance. And we do this with a relatively straightforward either gravity separation, or what I'm showing you here is a sieving technique where we separate greater than 200 micrometer fractions, a mid-range of 2 to 53 micrometers and a less than 53. And as you go down that, you increase the recalcitrance. What's quite handy about these is that these fit into some models quite nicely, and the sort of models generally look at fast, slow and passive pools and give some indication of their turnover time and the amount of carbon in each pool. The fast pool thought to be, it depends very much on the soil, to be frank with you, but in terms of the amount of CO2 coming off, the fast pool may have quite a significant amount. The slow pool, because it's quite large, sometimes can have a greater amount. It's the passive pool that's critical, though. That's real sequestration in that pool, because that contributes only 1% of evolved CO2 as very, very generic figures. And these fractionation models, physical fractionation and model pools are meant to roughly fit into some similar system. And there's all sorts of flaws and inadequacies about this, but it's a useful tool. We have some indication that there's good science behind this, because some of that particulate, this is NMR data, some of that particulate material has been looked at in detail, and we can see that those fractions are what we think they might be, at least to some extent. So here's some data that has used that sort of particulate analysis from one of the sites, from the Mediterranean Forest site, and here we can see, quite interestingly, that we have, let me just explain this to you first. Probably we have very superficial soil here, 0 to 2, 2 to 5, 5 to 10, and 10 to 20, and we're looking here at the accumulation of this labile fraction, the amount of carbon in this labile fraction in the surface soils. And this is what happens typically. We see here the early years, which we're excluding from our analysis at 2 years because the system hasn't settled down, and we have a native forest by comparison which might be deemed as some sort of target, and we've looked at whether we can show a significant increase across these data, and indeed we can. And wherever in this graph I show a solid line, there's statistical significance, and when it's dotted, that's just the relationship, but there's no statistical significance to that regression. So here we can see in the top 2 centimetres the most labile fraction increasing. Now all the data I showed you right at the beginning showing how you can increase carbon from crops to forest or from crops to pasture, this is the stuff that's accumulating. Nearly always this is the stuff that's accumulating. Does that matter? Well, it may very well, because it's also likely to go back to CO2 if the land use was ever to change. So there's a relationship here between land use and this sort of carbon. It's no more long-lived, or not much more longer-lived than the vegetation, and it's closely linked to the vegetation. Unusually, we've shown this intermediate level of carbon that's more slow to degrade. We found increases here, and that's unusual to find that, although not consistently down the profile. And then finally, with the recalcitrant stuff, the stuff we really would be nice to know how to increase this recalcitrant carbon. How do we increase the carbon that's going to hang around forever? Well, we know we can burn it, pyrolyze it in some way and create biochar, but you know, in terms of natural systems, how do we do this? We don't really know. Some evidence we have that this may be related to the huge range of carbon inputs we have from biodiverse systems, but that's very tentative at this stage. Carbon isotopes are also interesting. It gives us an indication of how much the vegetation affects the organic matter and the carbon in the soil. So here we have the relationship between delta 13 carbon and restoration age, which we're comparing with a number of native forest sites here. And we can see a general increase in the isotopic signature with time here. So what's going on? Well, these sites are dominated by C4 plants. And as natural succession processes continue, we find the C3 plants dominate the system, and that isotopic signature is reflected in the soils. This is the mineral soil carbon of the developing soils. Further isotopic evidence gives us some indication about how these soils might operate and whether they can recover in any tangible way. Can they self-organise in the way that natural systems purportedly do? Well, here I'm showing you the relationship between depths, A, B, C, D. We've shown these depths in a previous figure, and how they are nicely arranged here with more enriched at the surface and more depleted as we go through the depth profile. And that's what happens in the native soils. I've not shown errors on this, but this is replicated data. We're confident that this is so. And we can go and look at recently restored sites and do a similar analysis on the delta 13 carbon isotopes over the same period. And this is, I should say, is based on log carbon data to allow this relationship to happen. Nothing happens at all. No relationship for up to 8 years. But if we look at the system as it develops over further years, we see that relationship start to build. So there's some evidence that there's some self-organisation of those soils as the carbon isotopes start to reflect what's found in the native forest systems. We were also scratching our heads about how we can better identify and characterise and understand the changes that are happening with organic carbon. And so we hooked up with some organic chemists, and we tried to use a method that's based on accelerated solvent extraction, where we have a dichloromethane methanol extraction that's cooked up at high temperature and pressure, and we then use GCMS to try and resolve what's found. And we thought this is going to be fantastic. We're going to be able to identify a nice chronosequence progression through these soils, because people seem to have ignored this technique, although it's been around for some time. And then we realised why nobody did it, because we got 500 distinct peaks which left us kind of scratching our heads, thinking, right, how do we characterize these? But we started looking at it in a little bit more detail, and there's the native soils at the top, compared with years 1, 6, 12 and 17 in terms of chronosequence approach again. And we see here that actually there is some pattern perhaps that we can see as things start to change. Certain areas, for example, in the 17-year-old ones look quite different to both year 1 and the native soils. So we're seeing real differences occurring over time. So we looked at this on a multivariate basis. Now what I'm showing you here is a non-metric multi-dimensional scaling plot, which is quite different from a principal components analysis. To give you an indication of what this sort of software does, if you take a map of Canada and you have the cities, and you produce a grid of the distances between the cities that you might find at the front of that atlas, and you put that data into this software, it will produce a map of where those cities are. So it's a very good 2D representation of multivariate data. And we found that all of these years were separating off differently, and in fact the native system was quite different from the 17-year-old system, which was actually not that different to the 1-year-old system when we look at it in this multivariate way. And interestingly, and this is the only floristic data set I'm going to show, we found oddly similar findings for the trajectory of the vegetation over this period. That's not projecting too well here, but the point I want to make here is that the native system that appears in the green there is quite different from the 15-year-old blue dots over there. Thank you. These 15-year-old blue dots and the native ones are very, very far apart in terms of the floristics. So as the floristics have changed, so we see the same changes in the organic matter overall. Indeed, a vector analysis of what's going on with the species shows that one very weedy species, Acacia celestrifolia, is dominating this change. This is probably related to over-fertilisation. So finally, I just want to say a few words about microbial communities and carbon utilisation. And there's a whole range of ways we can assess soil microbial ecology, and we can measure functional diversity. Don't worry about all the details there from my colleague Jim Harris. I promised I'd mention his name to him, you see, who published a really nice little article in Science explaining why the microbes are so important in these systems. But they can help diagnose the quality of reclamation. Why functional as opposed to structural diversity? Well, all microbes may not be active in the structural analysis we do. We're interested in what they're actually doing from a functional perspective. And a report that my colleagues Carl Ritz, Jim Harris and others did looked at a range of different methods that could be used and found that community-level physiological profiling, or multiple substrate-induced respiration, which uses living organisms in the soil and exposes them to a range of substrates, and we can then monitor how well they're utilising those substrates through CO2 respiration, gives us the best idea. So here we see a range of, actually that's making me quite hungry just looking at that, I'll move on. But a range of substrates. My carbon utilisation efficiency, as you can see, is quite good for just about anything, but in the soil we have various specialisms in there, and some organisms can turn over some substrates, carbon-based substrates, better than others. So, and here's an example of some of those guilds of carbon substrates used. We can introduce up to 95 of them using a system called MicroResp, which is now a commercially available one from our colleagues at the Macaulay. Actually, I'm out of date. They're not called the Macaulay Research Institute anymore. They're now the James Hutton Institute. The James Hutton Institute, and paper published in Applied Environmental Microbiology. The driving force behind it was a friend and colleague, Colin Campbell, who works at that institute. And a very small amount of soil is introduced here, and we measure the response of CO2 flushing from that soil in a multi multi-titer plate reading scenario. So we can assess the whole population, and as I said, you can use up to 95 different carbon-based substrates. So it gives us a powerful tool for actually understanding how carbon is processed in these tools and how the microbial community responds to them. Here's just an indication of some of those substrates, and the ones we use, the data I'm just about to show you, is based on this paper by Brian E. Layler, who optimised a number of particular substrates to use particularly for these soils. And we're back at this, back at our mine in the Mediterranean, where we really wanted to analyse whether this chronosequence progresses towards native Jarrah forest system or not in terms of its microbial community, testing particularly the hypothesis that microbial function will develop towards that native forest. That's what we would like to see, of course, but we've no idea what we would find. And I've mentioned time for space substitution before, so I won't dwell on that one. But again, we had this chronosequence approach that we've taken before. So we hypothesise what we might see. We might see no effective response at all, no trend. And our multivariate, non-metric scaling plot may show no effect at all. Those different colours representing different chronosequence time points. It could all be a big muddle and a big mess, in which case I probably wouldn't be telling you about it. We could have found a direct trend where the colour here denominating a different time period succeeds one after the other in terms of the community in some way towards a native system. Wouldn't that be nice if it went towards a native system? So we have that. And then we have the other hypothesis, which was a reverse bottleneck. And does this reverse bottleneck not only show some progression towards an endpoint, but an increase in functional diversity as well as some sort of successional change towards an endpoint? So what did we find? Well, here I'm just looking at the native sites. We're not going to get very far if all the native sites that we analysed were different. And in fact, we found all the native sites were the same. There's no difference between them, which was reassuring. We then looked at the native sites versus recently restored sites, where we could look at, and we can do pairwise tests here. Year 1 versus native 1, year 2 versus year 1 versus native 2, and so on. And we can see where they're significantly different. The percentage levels I'm showing here, 5% is a significant difference, so the equivalent to P 0.05. So in this case, you know, they are significantly different between the first year and the native sites. There are already different communities existing. But here we're looking at the whole data set, and what does that look? What does this actually show us? Can we start to interpret the data that we've received from these multivariate profile responses of carbon substrates? And we do indeed see a reverse bottleneck, so an increase in the diversity of function through time. And we can look at the early years, and we can look at the later years, and we can actually do analysis between the late and early years, in all, pairing all cases, and in all cases they are significantly different. So we know that there is a time series going on here, but between the beginning and the end, they are significantly different, no question about it. And we can look at the old years versus the natives, and the old years are now not significantly different from the natives anymore. So we have had a microbial community response that is approaching that of the native soil systems for their microbial communities. So, can we accept or reject our hypothesis? Functional diversity increases with time, but the trend differs in the from the native systems, and thus the development is not towards the same function necessarily. It's not exactly the same. We're not restoring exactly the same system, which is often why rehabilitation or reclamation is used, because sometimes it's hard to do that. but there's the same degree and diversity of function in these soils, which we were quite pleased to see that was heading in the right way for these sites. So overall, I'll just draw a few conclusions for you to wind up. The carbon is accumulated in soils, post-mining reforestation schemes, no surprise there. Much of that carbon may be labile and have a short residence time and rapidly oxidize to CO2. This is the problem with many carbon sequestration schemes in terms of recalcitrance of carbon. Some evidence that more recalcitrant fractions may be starting to accumulate in the surface, which is interesting, and we don't know why, and there's more work going on to understand that. The timeframes which may lead to the development of recalcitrant carbon remain unknown, and there may be feedbacks between developing vegetation community in the soils, whether it's isotope related, whether it's in terms of the ACE GCMS data I showed you. There are relationships between the two that I think are likely to be important. And microbial carbon utilisation profile development is towards the same degree and diversity of function as in the native systems. But we still have some questions. How long will it take for recalcitrant particulate organic carbon to increase concentrations? When will sufficient changes be reflected at depth? As I said, everything was very superficial that I showed you. Can the delta isotopes of carbon be used as some sort of indicator of spatial and chemical recovery of soil carbon? And does floristic diversity play a role in the nature and properties of carbon and microbial function? I think probably so, but we need more to demonstrate it. I need to acknowledge these people who have all contributed to a greater or lesser degree, in fact nearly all of them to a greater degree if I look at that list, and the people who funded the work. Finally, if the organisers will allow me, I will just make a comment that the mine closure conference that ran at Lake Louise 2.5 years ago now is running in Johannesburg 1st to 3rd of October. And there are a few flyers going about. And there's the website for those of you that want to look it up. Just Google Mine Closure 2014 and you'll find it. We hope to have an international audience and maybe some Canadians coming to visit us at the conference later in the year. With that, thank you very much.